Welcome back to another Sleepy Steve story. Before we drift off tonight, remember to subscribe to help Steve-O out, and I will keep making more sleepy stories to help you study gently, revise quietly, and settle your mind at the end of the day. Tonight we are moving softly onward through Unit 3, Area of Study 2, from the quiet green world of photosynthesis into the steady inner work of cellular respiration. In the last episode, we looked at how light energy can be transformed into chemical energy in glucose. Now we are going to follow that stored chemical energy into the cell, where it can be released in a controlled way and used to make ATP. As always, there is no need to hurry. Biology can feel crowded when it is full of pathways, arrows, stages, and molecules. But tonight, we will let the ideas slow down. We will take respiration one step at a time. We will notice where each stage occurs, what goes in, what comes out, and why the process matters to living cells. Cellular respiration is an energy transformation process. It is the process by which cells break down organic compounds, usually glucose, to release energy that can be used to produce ATP. The important idea is that cells do not simply burn glucose all at once. They release energy gradually through a series of enzyme-controlled reactions so that the energy can be captured in useful chemical form. A strong exam sentence might sound like this. Cellular respiration is a series of enzyme-controlled reactions in which the chemical energy stored in glucose is transformed into ATP, which cells can use for energy-requiring processes. That sentence is calm, complete, and useful. It names the process, the source of energy, the product, and the purpose. ATP stands for adenosine triphosphate. It is often described as the immediate energy source for cells. ATP stores energy in the bonds between its phosphate groups. When ATP is hydrolyzed to ADP and inorganic phosphate, energy is released for cellular work. This cellular work might include active transport across membranes, muscle contraction, movement of vesicles, synthesis of large molecules, cell division, or maintaining homeostasis. You can think of ATP as a small, rechargeable energy carrier. Glucose contains a lot of chemical energy, but it is not the form that most cell processes use directly. Cellular respiration transfers some of that energy into ATP, and ATP then carries usable energy to the parts of the cell that need it. Now let us settle into the main site of aerobic cellular respiration. In eukaryotic cells, most ATP production occurs in mitochondria. A mitochondrion has an outer membrane, an inner membrane, a folded inner surface called cristae, and an internal fluid-filled space called the matrix. The folded cristae increase the surface area available for reactions involved in ATP production, especially the electron transport chain. The structure of the mitochondrion matters because each part supports a different stage of respiration. The matrix is where the Krebs cycle occurs. The inner mitochondrial membrane is where the electron transport chain is located. The intermembrane space between the outer and inner membranes helps build up hydrogen ions, which are then used to drive the production of ATP. Before respiration enters the mitochondrion, however, it begins in the cytosol. The first stage is glycolysis. Glycolysis means splitting glucose. It occurs in the cytosol of the cell and does not require oxygen. During glycolysis, one 6-carbon glucose molecule is broken down into two 3-carbon molecules called pyruvate. A small amount of ATP is produced and reduced coenzymes are formed. For VCE biology, it is useful to remember glycolysis as the entry point. Its location is the cytosol. Its main input is glucose. Its main output is pyruvate. It also produces a small net gain of ATP and reduced NADH. If oxygen is available, pyruvate can move into the mitochondrion for the next stages of aerobic respiration. A careful exam response could say that glycolysis occurs in the cytosol and breaks glucose into pyruvate, producing a small amount of ATP and reduced coenzymes that can carry high-energy electrons to later stages of respiration. This kind of sentence shows that you understand both the immediate products and their role in the whole process. After glycolysis, the pyruvate molecules are transported into the mitochondrion. Before they enter the Krebs cycle, they are converted into a 2-carbon compound. Carbon dioxide is released, and more reduced coenzymes are formed. This transition step links glycolysis to the Krebs cycle, and it helps prepare the carbon compounds for the next stage. The Krebs cycle, sometimes called the citric acid cycle, occurs in the matrix of the mitochondrion. In this stage, carbon compounds are broken down further. Carbon dioxide is released as a waste product. More reduced coenzymes, including NADH and FADH2, are produced. A small amount of ATP is also formed. The Krebs cycle is not mainly about making large amounts of ATP directly. Its major importance is that it produces reduced coenzymes. These reduced coenzymes carry high-energy electrons and hydrogen ions to the electron transport chain. In other words, the Krebs cycle prepares energy for the stage where most ATP will be made. So let us make that quiet link again. Glycolysis begins the breakdown of glucose and produces pyruvate. The Krebs cycle continues the breakdown of carbon compounds and releases carbon dioxide. Along the way, reduced coenzymes are produced. These reduced coenzymes then deliver high-energy electrons to the electron transport chain. The electron transport chain is located on the inner mitochondrial membrane. This is where most ATP is produced during aerobic cellular respiration. High-energy electrons from reduced coenzymes move along a chain of proteins embedded in the membrane. As the electrons move through the chain, energy is released. That energy is used to pump hydrogen ions into the intermembrane space. As hydrogen ions build up in the intermembrane space, a concentration gradient forms. The hydrogen ions then flow back through a protein channel called ATP synthase. The movement of hydrogen ions through ATP synthase provides the energy needed to combine ADP and inorganic phosphate to form ATP. This process is known as oxidative phosphorylation. Oxygen has a crucial role at the end of the electron transport chain. Oxygen acts as the final electron acceptor. It accepts electrons and hydrogen ions to form water. Without oxygen, the electron transport chain cannot continue functioning properly. If the chain stops, reduced coenzymes cannot be reoxidized efficiently, and the cell cannot produce large amounts of ATP through aerobic respiration. This is why aerobic respiration produces much more ATP than anaerobic fermentation. Aerobic respiration uses oxygen and can fully break down glucose to carbon dioxide and water, allowing a large amount of ATP to be produced. Anaerobic pathways do not use oxygen and release much less energy from glucose, so the ATP yield is far smaller. The overall word equation for aerobic cellular respiration is glucose plus oxygen produces carbon dioxide plus water and energy in the form of ATP. In symbols, it is often written as C6H12O6 plus 6O2 produces 6CO2 plus 6H2O with energy released. For exams, remember that the equation is useful, but it is not enough by itself. You need to explain the process as a sequence of stages. Now let us compare the stages gently. Glycolysis occurs in the cytosol, does not require oxygen, breaks glucose into pyruvate, and produces a small amount of ATP. The Krebs cycle occurs in the mitochondrial matrix, releases carbon dioxide, produces reduced coenzymes, and produces a small amount of ATP. The electron transport chain occurs on the inner mitochondrial membrane, uses electrons from reduced coenzymes, requires oxygen as the final electron acceptor, and produces most of the ATP. That three-part structure is one of the most useful ways to organize your thinking. If a question asks you to explain aerobic respiration, do not give a scattered list. Move in order. Glycolysis, Krebs cycle, electron transport chain, cytosol, matrix, inner mitochondrial membrane, glucose to pyruvate, carbon dioxide and reduced coenzymes, then ATP production with oxygen as final electron acceptor. Now we can look at anaerobic fermentation. Sometimes cells need to produce ATP when oxygen is limited or unavailable. Because glycolysis does not require oxygen, it can still occur. However, without oxygen, the electron transport chain cannot operate effectively, so cells need another way to recycle reduced coenzymes and keep glycolysis going. In animal cells, such as muscle cells during intense exercise, pyruvate can be converted into lactate. This is lactic acid fermentation. It allows glycolysis to continue producing small amounts of ATP when oxygen supply cannot meet demand. The ATP yield is low, but it can help cells continue functioning for a short time during high-intensity activity. In yeasts and some plant tissues, pyruvate can be converted into ethanol and carbon dioxide. This is alcoholic fermentation. It is important in processes such as bread making and brewing. Again, the key biological purpose is not to produce ethanol for humans, but to regenerate coenzymes so glycolysis can continue in the absence of oxygen. A useful comparison sentence might be this: aerobic respiration produces a much higher yield of ATP because glucose can be more completely broken down using oxygen as the final electron acceptor, whereas anaerobic fermentation relies only on glycolysis and therefore produces far less ATP per glucose molecule. That sentence gives the reason, not just the result. The rate of cellular respiration can be affected by several factors. Temperature matters because cellular respiration is controlled by enzymes. If temperature is too low, enzyme activity and reaction rates may be reduced. If temperature is too high, enzymes may denature, changing the shape of their active sites and reducing their function. Glucose availability also matters, because glucose is a major substrate for respiration. Oxygen availability is important for aerobic respiration because oxygen is needed as the final electron acceptor. The energy demands of the cell also matter because active cells require more ATP. It is also useful to connect respiration back to photosynthesis. Photosynthesis stores energy in glucose, using carbon dioxide and water to produce glucose and oxygen. Cellular respiration releases energy from glucose, using glucose and oxygen to produce carbon dioxide, water, and ATP. The products of one process can be the reactants of the other, but the processes are not simply opposites. They occur in different organelles, involve different stages, and have different purposes. Photosynthesis occurs in chloroplasts and transforms light energy into chemical energy stored in glucose. Cellular respiration occurs partly in the cytosol and mostly in mitochondria, transforming chemical energy in glucose into ATP. One stores energy, the other releases usable energy. Together, they help explain energy flow through living systems. There are a few common exam traps to avoid. Do not say that respiration creates energy. Energy is transformed, not created. Do not say that oxygen is needed for glycolysis, because glycolysis can occur without oxygen. Do not say that the Krebs cycle produces most ATP, because most ATP is produced through the electron transport chain. Do not forget to name locations, because location is often part of the expected answer. Another common mistake is to describe ATP as energy itself. ATP is better described as an energy-carrying molecule or immediate energy source. When ATP is broken down to ADP and inorganic phosphate, energy is released for cellular processes. This wording is more precise and more useful in biology. So, if you are answering an exam question, try to build a calm chain of cause and effect. Glucose is broken down. Electrons are transferred to coenzymes. Reduced coenzymes deliver electrons to the electron transport chain. Electron movement helps pump hydrogen ions. Hydrogen ions flow through ATP synthase. ATP is produced. Oxygen accepts electrons and hydrogen ions to form water. Each sentence moves the process forward. Let us now draw the episode gently together. Cellular respiration is the controlled release of energy from glucose to produce ATP. It begins with glycolysis in the cytosol. If oxygen is available, pyruvate enters the mitochondrion, where the Krebs cycle and electron transport chain allow much more ATP to be produced. Oxygen acts as the final electron acceptor. If oxygen is not available, fermentation allows glycolysis to continue, but with a much lower ATP yield. And that is enough for tonight. We have followed glucose from a stored energy molecule into the quiet machinery of the cell. We have watched it pass through glycolysis, the Krebs cycle, and the electron transport chain. We have seen ATP produced, oxygen used, carbon dioxide released, and water formed. And we have taken a pathway that can feel crowded and let it become slower, calmer, and clearer. Sleep well. Let the mitochondria keep working quietly in the background. Let the details settle gently. And when you come back to this tomorrow, the pathway may feel a little more familiar, one calm stage at a time.